going. Good morning. Good morning, good morning, good morning. For those of you who are here for the music as we waited for class to start, I the Mountain was singing The Boat, which of course the refrain is we're halfway there, and we're halfway there. This is kind of bananas to me, but congratulate yourself. You're halfway through your term. Hooray! And today, I hope you've brought your appetite because we are talking about feeding. We're talking about feeding today and Wednesday and next Monday. We've got kind of a tranche of, let's call it pie. The pie that we're eating today is talking about deposit and suspension feeding. But let's get underway. I will thank you. Well done for finding all those. I was saying as the music was playing that I'm not very good at all at the angular ones, but well done you. So it's been a while. I hope your Thanksgiving was good, and I hope it was all the things that you needed to be. When we were last here, we were talking about all this kind of thing. We were talking about essentially it sits under the umbrella of how hard it is to live terrestrially. It wasn't entirely terrestrial. In fact, that example that's down there is about a marine snail. But let's talk about it as temperature, effects of temperature on biology. So we talked about CT max and how to measure it, and the nuances associated with those measures, and what they might, strengths and skills or powers they might give us in terms of dealing with the climate crisis and predicting winners and losers in this battle that is going to be for the rest of our lives. But today we're going to change gears slightly, like I said, and we're going to change gears for the next few weeks to talk about feeding modes. And as you can imagine, when we're talking about invertebrates, we're talking about all of the modes of feeding. And we're even going to put some aside. I'll tell you about phylogenetic breadth that we're going to cover in today's lecture for one. But the strategies we're going to talk about are deposit feeding, like worms, earthworms, echurans, spoonworms, and suspension feeding, like krill, like barnacles, like sponges don't do that. They're kind of more of the flagella. But we'll revisit the myriad of strategies. And my goals for you, for us, for this time together, is that you can take these examples that we talk about in lecture and then move out into the world and interpret, synthesize, and interpret and synthesize when you run into new examples about how these fit within a phylogenetic context, within an ecological context, within an evolutionary context. And speaking of the phylogenetic context, it's a breadth. We got a big, wide swath of the Dunn et al. Phylogeny that we're going to be talking about today. We've got examples, arthropod examples, mollusk examples, annelid examples, cyclophorine examples. We haven't, you might not even remember that phylum. That's one of the 34 animals that we're going to be talking about today. Urochordates, and echinoderms into our neighborhood, the Mr. Rogers deuterosome zone of the Dunn et al. Phylogeny. And we're talking about sponges. Now, and sponges have a different kind of a stamp there because even though there's a wide swath of, say, 8 out of 34, one of the common elements is that largely we're going to be talking about kind of restricting this discussion, with sponges as an exception, to animals with a gut. And so I thought we'll come back to each one of these kind of taxa later on. We'll come back to the sponges. We'll come back to all of these kind of wonderful things as we go. But first of all, I want to take you to one of, I think, it's a good shared starting point, but it's also kind of a ridiculous diagram of the invertebrate gut. This is it. If you've dissected it out of just an invertebrate, we've got this mouth up at the top, into a buccal cavity, into a pharynx. And just like whenever you hear the word mitochondria, what do you hear? Put it in the chat or just say it out loud wherever you are. It's the powerhouse of the cell. And wherever you hear pharynx, what do you hear preceding it? It's muscular. You never hear of a limp and flaccid pharynx. This is a muscular, strong pharynx. It's going to break up materials that you're eating. Leading into an esophagus, a crop, potentially adding some more kind of physical breakdown of the materials that you're going to consume and trying to extract the nutrition from those into the stomach, into the digestive diverticula, like the Malpighian tubules and all those other things we've talked about, but this is a stylized one. an intestine, and rectum, and then out the anus. So we've got a foregut, a hindgut, and a midgut, thankfully placed in the middle. And this is essentially the model, the evolutionary model. As soon as you're into the bilaterium, this is a gut, right? And if you think back to 2700 last year when we talked about the mystery of the purple sock and about the first evolutionary appearances of a through gut, that's essentially what we've got diagrammed here. And there are specializations to different feeding types that we'll call on in the next little kind of 3 lectures together, or next 3 hours together, thinking about these things. But just as a shared map of the regions that we're talking about. So those are the interior regions, and then we'll talk about different strategies and different morphological and physiological adaptations that organisms have on the outside to enable their fabulous feeding. So today, deposit and suspension feeders. Wednesday, grazers, browsers, hunters in symbiosis. And then next Monday, a week today, we will dig down into the dirty parasites. Now, contemporary importance of deposit feeding. I think one of the coolest things about the deposit feeders is if you were to kind of just look at our planet laid out, and then remove all the ocean, how catastrophic would that be? Like 70% of the globe, no longer covered by seawater. But then you could get out your binoculars from your, unfortunately, very highly placed satellite, and you're looking down at this newly vacated ocean, formerly marine environment. You're looking at all that sediment. And for you, with your terrestrial biases, this is the first time you've really appreciated the fact that all of that ocean, all of it, as far as the horizon can see and farther and farther and farther, is covered with fine-grade sediments. All of it. So 70% of our planet is covered in fine-grained sediments, not excluding the terrestrial environment. Now and most of that sediment is in the deep ocean, not even in like easy to get to ocean. And so one of the consequences of this distribution of habitat is that this is one of the most frequently observed feeding strategies. And in fact, it's one of the oldest ones. So when we think about deposit feeding, so these feeders that are burrowing into tubes or crawling or wriggling across the surface, they're depending on some kind of a rain of detritus from above and microbes living in that sediment on the bottom, and then they kind of munch through and they extract their nutrition from the sediment, from the vastly overwhelming proportion. So what they're taking in, if it's if it's this much, it's a tiny fraction of that that's going to be nutritious. So the benthic habitat below the photic zone is where many animals first evolved, and it's one of the most abundant habitats around our planet. So ingestion of organic material that's in the sediment as food. And they subsist on a remarkably poor food source, sediment, delicious sediment. Nobody ever talks about sediment Saturdays or sediment Sundays to go along with Taco Tuesdays. Sediment's hardly ever excited anyone at all. And nearly all of it passes through that alimentary canal, that invertebrate gut that we drew out here on the left-hand side. Without being interrupted. So there's an immense amount of sediment that's taken in and an immense amount that comes out the other end. And so we have in this kind of top 2 to 10 centimeters, just as we think about the kind of the leaf litter and the first levels of soil, and probably as a Guelph student, your training has included some soil biology. But we have these deposit feeders that are subsisting on bacteria, benthic algae, grasses, but feces and detritus that is rained down from above. And that is an enormous part of maintaining, integrating, and maintaining this kind of linking the photic and the benthic zone. So here's an example of it says non-selective, but you can see the kind of the arms of this sea cucumber mouth parts pulling out and being kind of somewhat selective. Let's call it a little moderately selective. But it's ingesting these particles, the sediment, to get at the bacteria, the partially decomposed organisms, and other smaller protozoans that are living on top of that, like your amoeboids and your ciliates and your flagellates and nematodes, even mites, that kind of a thing. So a huge number, like pause just to say, from a phylogenetic perspective, their diet is broad. It's barren and subsistence and hardly ever exciting, but it is phylogenetically very, very broad. So what we see here is diagram this kind of feeding. We've got another holothurian. This is a synapta from the text. The diagram that's looking at, and it's pulled out. I've pulled out from another text that you can get to through your library by that QR code. Looking at the oral tentacles of either polychaetes at the top or these echinoderm, these holothurians on the bottom, that bury themselves in the substrate and then reach out. And so they're pulling from the detritus that's falling down and is sitting on the top levels of that kind of 2 to 10 centimeters. So some amount of particle selectivity, as you saw in the last video with the fingering kind of tentacles of the oral tentacles of the sea cucumber. They digest the organic material and then expel the inorganic material. Scaphopods is a mollusk example. This was the tusk shell that you might remember from 2700. And if your background or where you are from in the world includes coastal regions, maybe you've seen scaphopods. But for lots of people living in the center of the continent, they're one of the less frequently thought of molluscan lineages. But we've got these captacula, and this captacula is actually involved in bringing in food into the mouth. And so you see the arrows working here in this kind of bringing food in somewhat selectively, taking organics in from the sediment and feeding them via these ciliary tracts, running them down and along and into the mouth. And when you pull a tusk shell out, you can kind of see some of its burrowing head and then some of the captacula moving it around and searching around for organics in that sediment. Sipunculans are a kind of annelid where their organic material is being, again, pulled in from the background sediment, and they, like all other deposit feeders, have to solve essentially the problem. If your food source is so dilute, how do you possibly subsist? How do you eat enough to, in that kind of, remember in our life history, this lectures we talked about life history strategy, when we think about that, there's a limited amount of energy for any of the things that you need to do in the world, subsistence, metabolism, reproduction, that kind of thing. How do you get enough? Well, in this case, their solution is to eat an enormous amount of sediments. And sorted through the organs, the internal organs of that alimentary canal to get enough food. So the guts of deposit feeders, in that diagram that we put out a few slides ago on the left-hand side, they're typically unspecialized, and if there's one common kind of prediction, it would be that they have a long intestine. They have a very long, you'll see it kind of coiled up within their body. And so from a ecological perspective, one of the predictions would be that the density of deposit feeders across the world should be limited by food availability. That if your strategy is really just, I'm going to eat more, at some point, the amount of food, the amount of nutrition of organic material that's in that sediment is going to limit who's there. And so we're going to introduce a phrase that I'm sure you've bumped into in ecology and other classes before, and we'll unpack it further on Wednesday. But the idea of optimal foraging. Optimal foraging theory, kind of grown out of biology in the US and England in the 1970s, essentially tries to take a very reductionist view that pulls on lots of the life history theory that we introduced earlier in September. But it predicts that the food items that an organism will choose if it's going to maximize its net energy gain per unit time feeding. And so often it's thought about in terms of, well, how long does in ecology, maybe like, when does the lion look to the next gazelle if this one's taking too much time to eat it? Or the wolf begin to overlook a caribou to go for another one. Well, the strategy or the theory is useful because it's actually useful for the much wider variety of taxa that are included in our polyphyletic course in the invertebrates. So my question for you is, if deposit feeders, if this particular bristleworm, the Pygosio elegans here that's pictured there, if it's feeding optimally, what trend would you expect to see if you were going to look through the gut contents and the expelled frass, the poop at the end, for smaller particles? What proportion of them would be small particles, and the small ones being the organic ones, the more valuable one, and maximizing their net energy? So sketch out there in this kind of two things. So not a line here, but a bar. What are we looking at? Two, is there more small things in the gut, more small things in the sediment? Can't annotate. Oh, that's weird. You should be host tools. Annotation should be there. And if you want, Olivia, use the words. Put them out there. Nice. Thank you. Awesome. All right. So I see there's some that are predicting smaller, more valuable particles in the gut, some that are predicting smaller, more valuable particles in the poop, in the sediment, and some that haven't finished yet. Now, and well done. So well done. Participating in these kinds of things is I'm going to reiterate it. We just know it from studying learning. This is a low threat, low consequence kind of a way. This is a way of kind of being uncomfortable, answering a thing, and then moving on. And then if you see it again, you synthesize faster. So in fact, so well done you for participating. Right or not does not matter at all. So I'm going to take those, so let's say a 50-50 split-ish. I'm going to take these off. Thank you for participating on screen and in the chat. Clear all of those. And say, essentially, that if the prediction would be, and we'll unpack, like I said, optimal forage theory further on Wednesday. But the prediction would be that if the deposit feeders, if there's method to their madness, we should see more of the valuable sized particle retained in the gut and fewer of them expelled out into the sediments. Okay. There's my reminder to check the chat. I don't think I've missed anything. Throw it up again or rephrase it, and I will find it again. Now we're going to move on to some deposit feeder taxonomic examples. Now some of them we're going to move through quickly because you've seen them a lot of times. So it's really just a tip of the cap and a reminder that when you've been looking at the Lumbricus terrestris, at the classic model of that everybody's kind of pinned out and dissected either as a poor unfortunate earthworm itself or as a cardboard model or both last year in 2700 and or in high school, you were looking at a deposit feeder where the alimentary canal, the digestive system here, has a lot of adaptations, the muscular pharynx, the crop, the gizzard, all of these for dealing with the fact that the earthworm is essentially taking in the soil and trying to extract these things out of it that it needs. And there's some of the highlights. We've got an esophagus, we've got the gizzard, the crop, And then we've got these calciferous glands. The calciferous glands nestled, those 4 golf balls in the esophagus, are thought to release calcium carbonate to get rid of excess calcium from the body, because especially in calcium-rich soils like we have around here, in sedimentary or limestone kind of areas, to get rid of excess calcium. The gizzard grinds the food, and other glands in the intestine down here on the bottom release fluids to aid in digestion. Now, the calcium is kind of an interesting element. The calcium carbonate, those glands, we'll go back for a second, right up here, these calciferous glands that are releasing calcium carbonate to get rid of the excess. The rates of production probably increase with temperature, and that has to do with this ectotherm working at a higher metabolic rate. And that has consequences for how we model carbon sequestration, terrestrial carbon sequestration. And the calcium carbonate production in earthworms is probably related to pH and to tissue fluid, and has, like I said, to do with the high varying amounts of carbon dioxide or carbon in the soil. And this has been thought about since not just contemporarily, when we think like carbon sequestration being kind of a modern scientific, a contemporary scientific term, but it's been done for a long time. If you ever go to Down House, in where Darwin and Darwin's family lived when he returned from his voyage around the world in the Beagle, lots of the science that he did in Down House was about worms, and you can go and see the layout of many of his experiments that looked at the movement of soil, and he did some kind of very introductory calculations about how many worms, how much soil over this much of an area, that kind of a thing. But since the lifespan of the granules that are expelled can be hundreds of thousands of years, the worms can contribute to long-term reservoirs of calcium in the soil. And so we think about earthworms as novel arrivals to this fauna in this area, and we think about them kind of decomposing very quickly. Leaf litter that used to exist for weeks and years longer, but there may be a kind of an added side benefit with regards to carbon sequestration. The con is that in an area like we would have had here without worms on the left-hand side, the leaf litter exists as a habitat, as an ecosystem for a much longer period of time. This is a sped-up diagram of one on the right-hand side, the area, the terrarium, with worms, where they're completing that bioturbation. This is 15 weeks and 15 seconds. Now, if we move from a terrestrial example back into the marine to one that we've talked about already, and we think about sea cucumbers as deposit feeders. They're eating algae and invertebrates and waste particles, and they've got these oral tentacles that trap particles in mucus-covered tentacles that are pushed down into the mouth to ingest the food. And some species, as you remember from 2700, they can eject that entire digestive system when disturbed and then grow a new set within a few weeks. And they are, so there you see, this is a long kind of a snake-like, doesn't that look at the very snake-like, but this is an elongate sea cucumber with its oral tentacles searching for food and selectively bringing in, or what did I say earlier, somewhat selectively choosing the particles to feed into that mouth. Now the amount that's a big organism, right? So the amount of substrate that they eat is enormous. And if you followed some of the suggestions, or you're doing it on your own, of watching some of those deep sea dives from the Sebastian ROV and the Falcor that are moving around the world to different places, one of the things about deep abyssal plains where all of these sediments live is that there are millions upon millions upon millions of sea cucumbers all over the world. And so the role they play in turning over sediment, in inhibiting the overgrowth of bacterial mats, enhancing aeration and cleaning vast amounts, is enormously important, because the amount of poop that they produce, as you're seeing right there, is enormous because the amount of food that they're eating is a very small proportion of what's going in the front end. But so they're like a Roomba that's cleaning the sediment on the bottom. And I introduced you to this study, the infographic from this study last year in 2700, because I love it because it ends up scientists calculating using drones to count the number of sea cucumbers over a reef and then calculating the amount of sediment that each one of them could consume and then averaging out and saying weighing and saying the total bioperturbation of all the sea cucumbers on that one reef would equal about more than 64,000 metric tons of poop, which is, and this is the part that I love, approximately the mass of 5 Eiffel Towers worth of poop. And so we don't even have the Celine Dion's to sing on the front of all of those at the start of the Olympics. This is an enormous role, an enormous contribution played by sea cucumbers to the cleaning of the vast 70% of the surface of the planet. Other ones, icurans, these are spoonworms like you've seen, so annelids, specialized and secondarily unsegmented. They're an enormous deposit feeder. This is one that's been pulled out of its little kind of a retracted burrow. There's the burrow with from your Brusquet text, and there's the picture of that kind of tongue, this flattened proboscis. And then the video is kind of the proboscis once it's disturbed. But that proboscis is laid out, flapped, and then kind of inches in, encourages sediment to come into the mouth like this. And so it's basically, literally, it's a come hither, come hither. So the particles, this is the movement, the big arrow is the movement of the proboscis, and the small arrow is the movement of the particles into the mouth. So the proboscis is ciliated, and that helps it walk along the surface into the mouth. And then there's mucus that's produced as well and moved along by the cilius to make sure that the cilia is, or the particles that are being moved aren't just going to fall off or kind of move away because of the disturbance underwater. There is my next reminder to check the chat, and I don't see any questions there. So we move into suspension feeders. I love this video. This is a barnacle. So suspension feeders, another of the most diverse and phylogenetically diverse, but also numerous and abundant strategies across the marine environment, the 70% of the globe that many of you wish you were in all of the time. So these are animals that are removing and consuming particles suspended in the water. They evolved first in the benthos, like the deposit feeders did, and then they moved up into the photic zone through evolutionary time along different evolutionary trajectories within different phyla. And it's occurred many different times. This is not a monophyletically distributed trait. Sponges do this, lophophorate phyla, mollusks, annelid, arthropods, invertebrate chordates, and this one, this is your barnacle. And another angle. So it needs to be a suspension or a filter feeder. You need to have the medium moving. The medium is still the message, as McLuhan said. So you need to either create current or you need to be in a current. You can see this is doing a bit of both if you watch some of the particles cycling around in that motion. And then you need a capture mechanism, some kind of a filter. And different, as you could imagine, across different evolutionary lineages, there's multiple strategies. You can have cilia that do it, you can have mucus and cilia that do it, or you can have acetos, like a net, a basket. And we'll talk about several of those examples here. So this is love me a bryozoan. So this is one of the lophophorate, like the brachiopod, the bryozoans, these kind of, so the ring of ciliated arms that contain coelom that are bringing food into the mouth, into the center, and then pooping outside of that mouth, as I believe you're going to see this bryozoan do here on the outside. But you can see as the camera or as the microscope moves in and out, the ciliated fringe to each of these arms. So they're creating a current. And part of that is that that current is shown in the drawing here in the center. So it's drawing it into the mouth and expelling it out through the arms. And so that's separating particles from the water, grabbing them by the arms, capturing them, and then transferring them into the mouth and ingesting them, being a suspension feeder. And so we see annelids that do it. bryozoans that do it, bivalve mollusks, brachiopods, so the nominally like mollusk looking, but a lophophorate completely independently. We see deuterostomes like the brittle stars and the sea cucumbers, and these are all using like a mucociliary filter feeding in burrowing or mobile, somewhat mobile in life. And so there's cilia that's creating the current, but then there's also the creation of mucus to keep and to join the particles and to aggregate the particles so they can be fed into the mouth. Now, different filter feeders can also be, this is a urochordate, so this is as close to us as we're going to get in today's lecture. But this is Ecopleura, which is a pelagic tunicate, a urochordate, where the illumination from the ROV is shining through this large mucus net, this house. And so that is all out in the water as it's kind of swimming around. There's the small body there with its small tail, and then it sends out this mucus house, and that captures and then moves those particles into the mouth. So filter feeding with the secreted mucus network is not restricted to the urochordates. You see it within some gastropod mollusks. Sea butterflies have a secreted sphere or a funnel of mucus. There are polychaetes or annelids. There's other kinds of mollusks as well, and tube-dwelling annelids. And so it's a very successful strategy to filter feed and to suspend and or suspension feed. And I'm using those two terms kind of synonymously. If we had more time, we'd kind of split them up, but for the purposes of our shared time together, we can consider them as synonyms. Now, acetos, so filtering based on setae system appears and is most beautifully diversified in the crustaceans. So we see it in those barnacles, we see it in the legs of krill, we see it in crabs, and we see it in different amphipods. And so they're eating, if you're going to make slice up the menu of the things in the water column that they're eating, the water column that has to be there and has to be moving, they're eating bigger chunks. They're not competing for the very, very small things that the tunicates might be eating. So cetos filters are generally coarser than ciliary ones, and they can trap only larger particles. Now, when we think about suspension feeding, I focused kind of a taxonomic focus, but of course one of the things to remember or to remind you of, because you're aware of it, because you're invertebrate enthusiasts, is that there's a different emphasis on these kinds of feedings in some taxa dependent upon their life history stage. So villager larvae, for instance, that we see across so many of the molluscan lineages, trochophore larvae, those are, even if the adult doesn't eventually use this kind of filter feeding, the species does because the larval stage does. And so we see across different scales, not enormous, but we see large feeding currents in these ciliary suspension feeders where there's downstream collecting, but the compound cilia are generating a flow that is, and remember with the cilia, there is a recovery stroke, right? Like you're in this low Reynolds environment. So just like as when you're paddling a canoe, your blade goes in and then you knife it forward. These also those individual cilia need to be coordinated so that they're creating a unidirectional flow that can move the current, not just kind of agitate the water willy-nilly, but move it into the mouth. And so here is an example of a larval stage, a trochophore larva, where you can see that mustache of cilia beating around the edge. And so this is a longer-lived pelagic larva that's feeding and kind of existing out in dispersing long distances and existing out in the water column for a long period of time. And then the Panaria larva or the Brachiolaria larva. Examples, these, I mean, this looks like hello earthling. This is just kind of an amazing, this is an echinoderm larva. So the particles are collected on the upstream side of the current generating band of cilia and then moved into the mouth. And Then we've got the kind of the Muppet-looking actinotroch larva of the pharonids that is a ciliary siving, and so as it moves around, you'll see the beating of the cilia around, and that beating of that stiff band is distinct. There's a feeding current and there's a moving current. So in the mouth, in this oral hood, there's the cilia that you can see right in the mouth, and there's the cilia here by the foot that are moving it around. Different rates, different goals. One of them is moving it, one of them is feeding it. So what are they eating? From barnacles to actinotroch larva to brachyolarian larva to trochophore larva, they're eating similar things. Organic detritus falling down from the photic zone, particles suspended in the water column, other organisms. Lots and lots of the plankton comes into. So while themselves being part of the plankton, there's size variation across the plankton, and so some of the smaller members, phytoplankton and smaller zooplankton, some of the protozoans are going to be food for these suspension and filter feeders. Many of them are sessile or sedentary, especially if they live where the water is naturally moving, and they also tend to they can make a living where the environment is nutritionally dilute. Now that's a challenge for them, a dilute environment. And so to concentrate it, they have, this again relates to the size of the setae, the size you saw in that Ecopleura, the tunicate, the little tiny, kind of like a mouse size, my mouse, not a, but a computer mouse sized diagram, and then a large balloon that would fill up the rest of the screen of this mucus net that it's setting out. So that's their attempt to concentrate a naturally dilute food source. And then they create that water current that either passes over the body or through the body surface to bring food to them. And so in a way that the deposit feeders are not, the density here of suspension feeders is more limited by space, because they need to fill that space, whereas the deposit feeders are really, there's a lot of things, but they're more limited by the concentration of the food in that in the substrate, in the sediment. My reminder to check in, and I don't see anything, so let's talk about some crazy examples. This is one of my favorite ones. It's a Pandora's box, because we start out with the mouth parts of this lobster. So this is a lobster living, I believe, in Norway, Nethrops norvegicus, that can be both a benthic predator and a suspension feeder. So it does a lot of different things, but it has these setose legs that are filtering coarse particles. Now on those setae, so this is in the phylum Arthropoda, right? It's a crustacean. This is a SEM, a scanning electron micrograph of one branch of one of the filaments making up one of the feeding appendages, one strand of the net. And so then naturally, then that net has all of these hairs in it. That's part of the filtering action, but it also has all these tiny little muppets, the letter F there. That letter F is a feeding stage of an entirely different phylum of filter feeder, of the Cycliophorans. So an entirely different phylum, and this one is called Symbiome Pandora. And we only know of this phylum, this entire phylum, because we tend to eat this crustacean. And so it's an entire phylum that lives in the mouth parts and is filter feeding, suspension feeding, at two very different scales. A very coarse scale for the host and a very fine scale for the commensal, for the Cycliophoran Symbiome Pandora. And there's a zoom in, but you can see tiny little filters. So this is a filter on a filter. And it's so like, it's like there's filters all the way down. There's just feeding stages upon feeding stages. So two different taxa there. So we've got Cycliophorans and arthropods working on two different classes or two different categories. But then classically, of course, we've got sponges, right? So this is the non-mouth part or non-alimentary canal. But this is the classic. You've got the little collar, and you've got the flagella that's setting up a water current, and it's bringing water in, and the trapped particles are moving down the collar and being absorbed by a food vacuole. and through choanocytes into the syncytium, and the syncytium, it's syncytial, it can become practically anything, but in this case it's just going to kind of metabolize and share that amongst the body of the sponge. And not just as the example that I'm going to pull on today isn't a SpongeBob kind of an example, but it's a glass sponge. Glass sponges, classically, as you learned in 2700, in the deep, deep abyssal zone of the ocean, where they set up these marvelous, marvelous structures that are projected up and off the sediment into the water column to help more current and more water move through that meshed network, through the vacuoles to feed, so that in addition to current being produced by the flagellum, the choanocytes, that it can then be taking advantage of natural currents and movement of the ocean. And so they're not very selective feeders, but they're incredibly, incredibly fine cleaners of water. Unpack this for a bit. So this is for a glass sponge, deep water glass sponge, that on the left-hand side, we've got the percent change in the constituents in things in the water during single passage through the sponge. So a negative value from here means that the sponge is finely filtering, so much so that not just protists, protozoans, but bacteria are being removed from the water column. And over here on the right-hand side, we have the percent change in things like ammonia, showing you that, yeah, they're actually, they are still getting rid of that metabolic waste. So just marvels of engineering, marvels of nature, and like so many biological marvels of nature, engineers of our own species have looked at these and thought, I bet you we have within the structure of a glass sponge, which comes up again and again, this kind of skeletal motif. That's probably not by chance. There's probably a reason for that. And their hypothesis was that it was one of the easiest ways to get water to move through a thing without there being a lot of shear forces on the structure itself that would tend to topple it. Sometimes the point of attachment of the glass sponge is it's much narrower than the feeding array that often kind of extends above it like a vase. And so biomimetic engineers, teams of engineers and biologists, were looking for a filter that could work very finely to help recover nearshore environments and from oil spills, which we tend to do. This is a problem with the Anthropocene. And one of the problems that they were having was that we can design a really good filter, but we tend to need very calm water for it to work in. And as soon as the filter is moving around, its efficiency goes way down. And so they essentially started to mimic the structure of the glass sponges. And what they found was that by mimicking the multi-layered skeleton, that they created a filter that would be, and this is supplemental material from their paper, where the filter is not essentially being moved. And so frequent collisions, deflections didn't matter. That the efficiency of the filter was just higher with this design than anything else that we had designed to that point. Now, other incredible filter feeders, suspension feeders that we've talked about already when we talked about the plankton, were the krill. And we tipped our hat to the krill and thanked them for their contribution to sequestering carbon, to feeding so many things, to changing the weather. And the part of the reason that they're doing that is because of this thoracopod. So these legs in here, see the legs at the back end of the abdomen, they're moving them around. But the basket-like set of legs, the foremost appendages, are used to filter the large amounts of food that they need out of the water. And of course, you've run into other filter feeders before when you've talked about and thought about mollusks. That when we think about bivalve mollusks, and this is a diagram that we're moving quickly through because you've seen it already in 2700, and all it's showing you is that inside of those valves, you have an inhalant current and an exhalant current, and then the gills are not just respiratory surfaces, they're feeding surfaces, because we are, as the gills are setting up currents. There is cilia that's moving particles either towards sorting them into things that we wish to eat and things that we wish to ingest as pseudofeces. So undesirable and desirable particles and currents being created by the cilia. And so different layers of cilia here. So this is a look at one different mollusk where we've got the lateral cilia here that are moving the water, and then the lateral frontal cilia that are up here, the kind of the like a gooseneck, I guess, but gorgeous, that are more, let's not call it a gooseneck, let's call it a harp, the kind of the lateral frontal cilia. And those are the ones that are capturing the particles in the mucus that are then moving them around. And as you know, especially if you live down the shoreline of Lake Erie, these filter feeders, the mollusks, are incredibly efficient. So this is an example here of, so they've got one half of a tank and a time series, and you can watch the turbidity decrease here. The clarity of the water increases. And this is essentially what happened in Lake Erie with the arrival of the non-endemic zebra mussel in the 70s and 80s, that the turbidity of the water, that highly productive most southern Great Lake, was just being pumped full of phosphates and was full of algae, and you couldn't see. It was just a soup, a green soup. And as soon as we had this kind of exploding population of filter feeding mollusks, the zebra mussel added to it, the lake has become clearer and clearer over your lifetime. Okay. So, invertebrate feeding modes. Today, we've talked about deposit feeders, and we've talked to some of the phylogenetically broadest and oldest strategies. Getting your food from the sediment or getting it from the medium in which you're moving and living and off the water. So whether it's aquatic or marine, but generally we've talked about marine examples today. So we talked about annelids, echinoderms, annelids again, mollusks, sponges, cichliophrans. Where are we going next time? We're going to talk about symbiosis and grazing and browsing and hunting. The labs this week, as a reminder, this is a help session, but you need to kind of monitor and create your own itinerary with Shayla. So she's written on CourseLink about how to do that, how to contact her to set up a time to talk about any questions that you might have. Another ongoing reminder that your the deadline for the second invertebrate R assignment in this examination into the deep thermal vents of the world is going on right now. My suggestion is leave it till the last minute. And then a final suggestion that you can go, and it's never too late to start grouping up and studying for the case studies because they're going to help you prepare for the final exam. And that's kind of it. I'm going to stop recording in a second. Thank you, and take care of yourself. Take care of others that are around you, and I will see you in a second, because I'm just going to stop the recording and hang out for some questions.